Hello everyone, welcome back to my channel Crack with Trick by Shiva. So today we are moving on to the next topic from the chapter Biotechnology Principles and Processes. So we were already discussed about the major concept in the chapter Biotechnology. Okay, I hope you have gone through it. Okay, now we can move on to the next topic that is actually processes of recombinant DNA technology. So already I have told you about the recombinant DNA. How to make a recombinant DNA? What are the major tools which are required for making recombinant DNA? So this topic and all we were already covered. Okay. So those who have doubt in that topic, they can go through the channel and the video is already uploaded. Now we can move on to the next topic that is actually the processes. See, the processes of recombinant DNA technology, it involves a few steps. Okay, first step is actually isolation of genetic material. Now the second step is fragmentation of genetic material by restriction endonucleases. Now third step is actually separation of desired DNA fragment by gel electrophoresis, and the next step is actually ligation of desired DNA fragment into the vector. Now the next one, that is actually introduction of vector into the host organism. Then after that culturing of host organism. Then the last step is actually extraction of desired product. Okay, so these are the major steps. So each of the step detailedly we have to discuss. Anyhow, first we can go through the first topic, that is actually the isolation of genetic material. See, as I have already told you that for making a recombinant DNA, okay, a DNA which is having more than one character or segments of other DNA, that is recombinant DNA. So for making a recombinant DNA, we should have to extract the DNA from a cell. Okay, so either we can use a plant cell, like for experimental material we can use many type of cells. Okay, so if we are using plant cell as an experimental material, I told you that genetic materials are present inside the cell, right? So we have to separate the genetic material from the cell. So separating genetic material means actually it is somewhat difficult because the cell is actually made up of cell wall. Okay, so a protective wall is there. So first of all we have to digest the cell wall. So if we are using a plant cell, see, it is actually the made up of, the wall is actually made up of cellulose. Okay, so we have to use a cellulase enzyme in order to digest the wall. Okay, now the next bacteria, it is actually peptidoglycan. So in order to digest the peptidoglycan, we have to use lysozyme enzyme. And if it is fungi, we know that it is actually the wall is made up of chitin. So chitinase enzyme we have to use it. Okay, so just imagine this is actually a cell. So we can see that inside the cell we can observe the genetic material. So genetic material we have to separate the cell. We have to separate the genetic material. Either we can use if it is a plant cell, as I have already told you, that cellulase. So just consider this as a plant cell. Okay, so we have to use cellulase and we should have to separate the DNA. Okay, the genetic material. Now the genetic material is actually intertwined with the other component like RNA, protein and other macromolecules will be there. So for separating the DNA from RNA, we can use ribonucleases. So ribonucleases will cleave the RNA and we can separate the pure form of DNA. Okay. Now for separating the protein, we can use a protease enzyme. Okay. So if the DNA is actually connected with any other macromolecules, we have to use appropriate enzymes for separating the DNA. Okay. Anyhow, after doing or after all this enzyme actions, we should have to add like chilled ethanol. So when we will add chilled ethanol, the DNA, it will be precipitated. The precipitated DNA, we can separate it. Okay. So these are the first or major characters in the first step, isolation of genetic material. Now, In the second step, that is actually fragmentation of genetic material by restriction endonucleases. So you just consider this as actually the DNA. So as I have already told you that after separating the DNA, we should have to add a restriction endonucleases. See, the restriction endonucleases, it will act on the DNA and it will make many fragments. Okay, it will make many fragments. Okay, so this is actually the second step, that is actually fragmentation of DNA by restriction endonuclease. Already we have discussed about restriction endonucleases. Okay, have you remember restriction endonucleases? It will identify the palindromic sequences and it will cut the DNA. Okay, so that we have already studied. Okay, so here, so this is actually the second step. Now the third step, that is actually extraction of desired DNA fragment by using gel electrophoresis. Okay, so we should have to separate this. Consider this is actually the fragment which we required. Okay, the desired DNA fragment. Now these fragment we have to separate it by using gel electrophoresis. Okay, so that is actually the third step, gel electrophoresis. So gel electrophoresis, it is actually one of the most important topic from the chapter biotechnology, principles and processes. So in the second step of process of recombinant DNA technology, I just mentioned that adding restriction endonucleases. So in the second step, when we will add restriction endonucleases into the DNA, the restriction endonucleases will identify the palindromic sequence and it will cut the DNA into many fragments. Okay, now in the third step, we should have to separate the smallest DNA fragment from the group of fragment. Okay, so just consider this as actually the DNA. So in this DNA, we will add restriction endonucleases. So it will identify or inspect the DNA and it will identify the palindromic sequences, make the DNA into small fragment or make the DNA into different fragments. Okay, now just imagine that, so from this we have to separate a small DNA fragment. Now for separating the small DNA fragment, we can use the technique that is actually, we can call it as gel electrophoresis. So gel electrophoresis. See, in case of gel electrophoresis, in gel electrophoresis, as we all know that DNA it is actually negatively charged. Okay, DNA is actually negatively charged. Okay, so this negatively charged DNA, we are forced to move towards anode under an electric field through a medium. Have you got it? So DNA is negatively charged. Now why it is negatively charged? Because of the presence of phosphate group that we have already studied. Okay, so this negatively charged DNA, we are moving or we are forced to move towards anode under an electric field through a medium. So the medium what we are using here is actually agarose gel. Okay, so the medium what we are using is actually agarose gel. So this agarose, it is actually a natural polymer which is extracted from the seaweeds. Okay, so agarose is actually the natural polymer which is extracted from seaweeds. So in this we are using agarose as a medium. So just consider this is actually the experimental setup. Just consider this actually the experimental setup. So this is actually the, here we have added the media. So the media what we are added is actually agarose gel. Just keep in your mind, just for understanding purpose. Okay, now here in this side, see in this side, so we will add the DNA fragment. Okay, in the well we will fill the DNA fragment. Okay, so as I have already told you that the DNA is negatively charged. So this is actually the anode end. So this is actually the cathode end. So the DNA, the negatively charged DNA, it will move towards the anode. Okay, the negatively charged DNA move towards the anode. Now the very important point is actually, see the smallest DNA fragment. Okay, so the smallest DNA fragment, it will move further than largest fragment. Okay, see, it is because of the sieving effect of agarose gel. See, agarose gel, the molecules of agarose gel are tightly packed. Okay, so the small fragment can easily passes through it than large fragment. So smallest fragment, it will be move further. See, as like this, the smallest fragment, it will move further. So this is actually the smallest fragment. Okay, so just consider these are actually the largest fragment. Here also the largest one, this is actually the smallest one. So as like that, so the smallest fragment, it will move towards the anode. Okay, so because of the sieving effect of agarose gel, the smallest fragment will move towards the anode. Okay, now so we don't know whether the extraction is happened or not. So for understanding that, we should have to add etherium bromide. Okay, so when we will add etherium bromide, so in etherium bromide, and after exposing the gel under UV radiation, we can see orange colored bands. Okay, here I have written green, but orange colored band we can observe in the gel. So that orange colored band indicating DNA. Okay, so after that, we can cut the gel and we can just cut the gel and separate the smallest DNA fragment. That process is actually called elution. So elution is actually the process of cutting gel and separating the smallest DNA fragment. Okay, so once again I'll tell you, in agarose gel electrophoresis, see, we as because of the negative charge of the DNA, or the DNA is negatively charged, which are forced to move towards anode under an electric field in a medium which we will call it as actually agarose. Okay, agarose is the medium. So because of the sieving effect of agarose gel, to the smallest DNA fragment, it will move further. Okay, and then after this, we should have to add etherium bromide and expose under UV radiation. We can observe a small or we can observe the bands, orange colored bands. Okay, so then after that, cutting the gel and separating the DNA fragment, that process is called elution. So these are the major topic in the gel electrophoresis.